So it's a great pleasure to be here. I said to Greg a few moments ago, we've come a long, long way. We actually had the very, very first meeting on GBA and Parkinson's disease at the NIH in about 2003 with about 20 people in a small room in Fogarty. I think maybe one or two people in the audience were there. Then had another one in 2007 when we began to get a momentum, and that's when neurologists began to get on board. And then in 2010, around the New England Journal paper, we had many more, and the field is just taking off. If you look at PubMed, the number of papers is rising really exponentially. It's really very amazing. So the first speaker today is Greg Grabowski. He's probably the only one here who's been in the Gaucher field longer than I have, and he will give you an introduction to the field.
All right, good morning. Happy to be here. It was an interesting day yesterday getting here, but we all made it. And since Ellen and Zev took about 7 minutes of my time, I get 7 minutes of time. I am a professor emeritus, as you see with there on the slide, and these are a few of the companies and people that I've worked with and work with now to forward this area and move it on. Now, I'm supposed to talk about wild-type GCase, and I want to make a couple of points here. GCase, or glucocerebrosidase, is an enzyme. GBA is a gene, and we have to clean up this literature. Unfortunately, glucocerebrosidase has the same EC number as glucocerebrosidase 2, which is a neutral is not a lysosomal enzyme. So they have the same EC number. So things can be confused, and we have to be specific. The important part of GCase getting to the lysosome was discovered a number of years ago, 1993, and it is dependent on lysosomal integral membrane protein 2. This is a mannose-6-phosphate independent sorting, so it's very different than many of the other lysosomal enzymes. The loss of LIMP2, a knockout in the mouse and in humans when they lose activity, it does not lead to Gaucher disease, so there must be some other mechanisms by which glucocerebrosidase gets into the lysosome independent of LIMP2. GCase binds to LIMP2 in the ER around neutral pH. It is released in the lysosome at acid pH, and so it's acid controlled. It has 5 glycosylation sites, 4 of which are occupied. Only N19 is the one that is absolutely essential for enzymatic activity. The other asparagines can be modified a bit, and enzyme properties change a bit, but they are still active. So glycosylation is important to the activity, but N19 is very important. Crystal structures. There are at least 30, potentially more, crystal structures for wild-type GCase. There is one mutant, N370S GCase, that has been crystallized, and it is fundamentally similar to, except for one little loop, the wild-type enzyme. What you see here in the center of the screen is the enzyme with its 3 domains, 1, 2, and 3, and a bunch of amino acids that are highlighted here. Those are to represent mutants, missense mutations that do occur in Gaucher disease, and they are spread all throughout the enzyme. There's no functional relationship for where they are and what type of Gaucher disease you have. So it's very different. The active site has been fairly well characterized. E340 is the nucleophile. E235 is the acid base and catalysis. It also has an anionic cluster binding site on the enzyme that we'll get to and why that's important for interaction with certain kinds of negatively charged phospholipids. The substrate it's always a question of how this gets into the active site. We've used a variety of inhibitors. The deoxynitroglycin, the N-alkyl deoxynitroglycins have sort of probed how deep you can get in. You need probably somewhere between a maximum of 9 to 12 in the acyl chain to have the most potency of attaching to the active site. Now, we all think about how these enzymes work, and I'm going to talk about activator proteins for a minute because they're really important. There are several different kinds. I'm going to talk about 4 in particular for a short time. Sapacin B is called a solubilizer. It's a small molecule. It'll go in and it'll pull out the substrate and hand it off to the enzyme. There are things called liftases, which is sapacin C, which we'll get back to, and sapacin D, which distort the membrane and present the substrate to the enzyme in a significant way, so it has better access to this, because these lipids are stuck in membranes. Glucocerebroside is not floating around in the lysosome. And if you think it is, you should look at a Gaucher cell and find out how insoluble it really is. The last one, sapacin A, works with the enzyme in Krabbe disease, and it is a dimer. It forms a total tetramer with that enzyme to complete the active site. Now, this is how this goes. It has to be thought about when we think about mutations here. Once this was cloned, the prosapacin molecule was cloned by us and others, there were 4 sapacins that were encoded. They are very highly homologous. Their structure is very similar. They're all about 80, 83 amino acids in length, and they are processed in the lysosome to individual sapacins. There's a major pathway, and these become then monosapacins, and that's where they have their function in the active site. So this is a very important molecule because each of these sapacins controls the activity, at least in part, of a glycosphingolipid enzyme, and sapacin C is specific for glucocerebrosidase. But it gets more interesting and more complicated. This is prosapacin here, and this is another multi-component protein called progranulin. And it turns out that prosapacin and progranulin bind to each other in very specific residues, and then that complex is sorted to the lysosome and for processing. It also can bind differently to the mannose-6-phosphate receptor, and prosapacin binds to progranulin, and that gets sorted to the lysosome where they get processed too. That's important because each of these individual sapacins and granulins in progranulin have different functions, one of which is the section E of progranulin. This is, if you isolate this is blood-brain barrier penetrant. It binds to glucocerebrosidase. It enhances the activity of glucocerebrosidase in vitro and in vivo. And if you administer this to some mice with a point mutation and a progranulin deficiency, you fix the visceral and CNS disease. And that was published by us last year. In addition, those are mice, humans, that have L44P homozygosity, Gaucher disease, but also have had a knockout made of progranulin and N370S84GG. The enzymatic activity and a lot of the lipid abnormalities are corrected by adding this peptide in. So here's a basic science piece that suggests that there is a way to use another activator to move this along, and the wild-type enzyme responds to this very nicely. Then we come to phospholipids. And I don't know how many of you are interested in BMP, but bis-monoacylglycerol phosphate is a very important molecule, and it's not a single molecule. There are multiple ones. This is synthesized. The synthesis is cell and organ specific, and because it interacts with glucocerebrosidase, it alters the enzyme activity differently in different tissues and different cell types. It's made from phospholipid dehydrogenase 3, which is found mostly in microglia. So we got cell-specific expression leading to an effect on glucocerebrosidase. A deficiency of this enzyme leads to neurodegeneration and it's driven by microglia inflammatory effects. PLD4 is mostly in peripheral myeloid cells and organs, like, for example, the spleen, and a deficiency leads to a splenic glycosphingolipid disease with glucoceramide and glucosphingosine being stored or accumulating. This implicates BMP in the modulation of GCase function via the interaction with multiple anionic sites, the binding sites that I showed you on the surface. This has not been well characterized yet. And this is what the molecule looks like, and the isomers come from the different types of lipids that are attached to the head group here in BMP. It is found only in these intraluminal vesicles in the lysosome of any cell. That's the only place it's found. Now, and these are the models that have come out of this from a variety of different sources. And we'll look at this one over here, which is progranulin in it and in the lysosome, and you take a look at what happens. You have these intralysosomal ventricular structures, microglia or microspheres, and they have different populations of different kinds of lipids, different kinds of enzymes, and so forth. The pH in the lysosome, as you know, is somewhere between 4.5 and 5. Inside these vesicles, the pH is 7.2, sorry. And therefore, whatever activity is happening is not of these from, let's say, GCase, is not inside, it's outside these vesicles. Some of these can be led to secretion and so forth. And what you get is you get activation of glucocerebrosidase, activation of sapacin C, participation of some of the granulins, in particular E, from progranulin, and binding to BMP here and the substrate. Here they use glucosphingosine, but it could very well be glucoceramide. And similarly, you have this kind of interaction here without the presence of sapacin C. So you have intermediate kinds of activity, and we do not understand exactly how this is all working within the cells. If you have progranulin deficiency, you end up with accumulation of glucosphingosine and lack of substantial glucocerebrosidase activity. So we have multiple different compounds being used as activators. And just to show you this is really real, we're using a particular mouse model here with a progranulin deficiency and glucocerebrosidase mutation. You get accumulation of glucoceramide in various tissues, including the brain, depending upon the state of whether or not you have a combined deficiency or singular deficiency. And in the brain, it's even more important here, in the lungs, and in other tissues like the liver, it doesn't seem to have as much functional activity. The point being that all kinds of different tissues and interactions of this enzyme in various cell types leads to different manifestations of metabolism, including Gaucher disease. Now, it gets more complicated. Decreased glucocerebrosidase activity and stability alters not only lysosomes, but also the mitochondria. And decreased glucocerebrosidase function and structure leads to an increase in glucoceramide, you know that, and it disrupts the normal lysosomal structure and function. And it dissociates mTORC1 from the lysosomal cytoplasmic surface, leading to dysfunction of the clear system, which is a system that is a coordinated lysosomal expression and regulation system of transcription factors. And it alters the ability of mTORC1 to phosphorylate these or not. The wild-type enzyme also enters directly into the mitochondria. How that happens, I haven't yet gleaned from the literature, but it has stabilizing effects on the complex C1 function. So we're talking about respiratory chains here, and also reactive oxygen species functions. And the mutant enzymes may partially or fully lack these functions. They disrupt the C1 complex and ROS formation. The increased glucoceramide destabilizes the mitochondrial membranes and leads to altered mitophagy, leading to a leakage of double-stranded mitochondrial DNA to interact with C-gas sting, with activation of inflammatory pathways, with interferons, mTORC1 metabolism, with detrimental effects on neurons and neurodegenerative disease, including Gaucher disease. So this becomes a very complicated system, and I will show this slide instead of the last one just because this is a little more high level. Here's C-gas, and it binds double-stranded mitochondrial DNA, which then, because of the leakage of the mitochondrial membrane, leaks out and gets attached to sting, which enhances a pathway all the way to mTORC1, which sits on the surface, or not, of the lysosomal membrane. So, and there are other complicated components of this, but this is the mitochondrial component. And now we have GCase in the lysosome, we have glucoceramide on the lysosomal membrane, and we have it in the lysosome. So we have a multi-subcellular compartment disease process. And this is a summary of everybody's worried about modifier genes and how things can change in Gaucher disease. And here you have something quite simple in a sense. These entire pathways are modulatory pathways. If these two compounds, these two transcription factors, are phosphorylated by mTORC1 or not. If you lack phosphorylation, that happens when the mTORC1 is detached from the lysosomal membrane, and it enhances the catabolic pathway. When it is phosphorylated, these cannot enter the nucleus. mTORC1 is attached to the lysosomal membrane, and you enhance the anabolic pathways. Now, this is not an on or off switch. These are rheostats that you turn on or turn up a little, and things change on all these different pathways because of the function of glucocerebrosidase and probably other lysosomal enzymes. For sure, lysosomal acid lipase is involved with this. This was supposed to be simple. And the point about this is we still don't know, for example, the structure of glucocerebrosidase in the cell. In other words, we were talking about it before. Is it a monomer, or is it a dimer, or something else? And that makes a difference potentially in terms, my prejudice is it's a monomer, but if it's a dimer, and you have a mutant glucocerebrosidase and a wild-type glucocerebrosidase in a heterozygote, and they try to dimerize, it could be that that mutant glucocerebrosidase destroys the function of the wild-type and creates a new kind of gain of function, which you're going to hear about. You're going to hear about mitochondrial effects a little later in the program, too. I just want to thank all of these people and more over the years who have participated in these studies. The Project Hope Gaucher Disease Initiative has been an enormous impact in my life to see over 600 or so Gaucher disease patients, mostly with neuropathic Gaucher disease, and to watch the incredible heterogeneity in that particular disease, and then, of course, having to account for all kinds of other effects. And then to over 2,500 Gaucher disease patients and families who I've taken care of over the years. Thank you very much.
Anabolic and metabolic, right? And I was confused because I thought that when you increase autophagy, you need to increase autophagy and mitophagy in order to keep the recycling system, right? So I was confused by that because I saw the increased autophagy, well, maybe I'm reversing it, but basically it confused me.
Yeah, well, again, this is fundamentally a metabolic control system. a center the lysosomal membrane is. And the coming on or going off of mTORC1, I mean, is a huge, there must be thousands of articles in the last couple of years on this. And what it is it doesn't come off completely. It doesn't destroy mTORC1 function completely. It's a rheostat. You turn it a little bit this way and you disbalance
Yep.
catabolic and anabolic pathways, and then you have to rebalance those in a therapeutic response to an insult at the lysosomal membrane.
Okay, thanks. Thanks.
All right, I think we'll get the rest of the questions in the question period.